Thank you so much for this kind introduction. And hello, everybody. Good morning. My name is Pascale. I'm a postdoc in the lab of Ido Amit in the Weizmann Institute of Science. And I would like to talk to you today about cracking the code, understanding cellular communication in cancer using single-cell genomics. While I'm talking to you and my words reach your ears and might trigger a reaction in you, also the cells in our body constantly talk to each other. And this cellular communication is very important for basic processes such as the formation of a human body. It is also very important for the defense of our bodies from infections. So if somebody here in the room would sneeze, then the immune cells in the other participants here would need to communicate to each other how to defend the body from this virus that is in the room. If cellular communication gets out of balance, this can lead to the occurrence of diseases, such as, for instance, cancer or also autoimmune diseases. In normal states in the body cells would tell the immune cells, all is fine, you don't need to do anything. However, if a virus is there and infects a cell, the cell would tell to the immune cells, hey, I'm infected, please come here and kill me. And the immune cells would do this very happily, actually. In the case of cancer, this is a bit different. Even though the cancer cell is defective and it's duplicating like in an uncontrolled manner and it should be killed, it tells the immune cells, do not attack me, and even come and help to hide me. And this communication is of course not done with words and with ears as shown on the slide, but it is done by the expression of protein ligands and also receptors. The ligands can be just on the surface of cells, or they can also be released from the cell and secreted to the surrounding. The cells that have the receptors can get these signals and can then change their shape or function according to the signals they receive. In cancer, the cancer cells deactivate the immune cells and they even team up with some cells that help them to further grow and proliferate. One way of cancer therapy is called immunotherapy, and it's a therapy form that has the aim to reactivate the patient's own immune system to again fight the cancer. One type of this therapy is cytokines. These are small secreted proteins which are also usually there in the body and are like normal cellular communicators. But we can use them as a treatment to artificially reactivate the immune system. They then get activated again and can attack and eliminate the cancer. There is a huge interest in cytokines as clinical therapy for cancer, which is also represented by the huge amount of clinical trials on cytokine as cancer therapy. However, so far it is not yet so successful as only 2 cytokine therapies have been approved. And also many of the clinical trials fail due to the occurrence of toxicities or also just because they're not efficient enough. So why is it so difficult to make cytokine therapies? Imagine a tumor as a very full field filled with many different cell types. There are cancer cells, there are immune cells of various flavors, and also even more cell types that support this whole structure of the tumor. If you now just scream into this tumor, attack the cancer, this is very unspecific. Many different ears will hear it, ears that are in the pro and ears that are in the anti-cancer team. Furthermore, it can be too loud because the cells can not only attack the cancer, but even also the own body, which then can cause toxicities. So what the aim is to send a voice message only to very certain specific cells and locations. But how can we do this? We need first to answer some questions, like which of the cells should hear the cytokine words? Also, are there some locations which are better suited to be activated by cytokines? Then also, when immune cells enter this crowded field of the tumor, they start to change their shape and to get deactivated. We don't know when in this process, which we call cellular differentiation, it is best to activate the cells with a cytokine. And also lastly, there are many different cytokines. Which word combination is the best? This we also still need to find out. I am using mouse tumor models and also human cancer biopsies, which I treat with cytokines and cytokine combinations to answer these questions. I'm using a method or a group of methods called single-cell genomics, which allow to study individual cells. You have to imagine that before this method occurred, we had to study this whole field as one. So we couldn't really discriminate between the effects on individual cell types. With this method, we can study the genetic profile of each individual cell and how it changes after being stimulated with cytokines. I'm also using a method called spatial transcriptomics, where we, in addition, also can measure the spatial coordinates of the cells. And we know then from where exactly in a tissue each cell is. We also use a technique called Zman sequencing, which was developed by another Azrieli fellow, Daniel Kirschenbaum, which allows us to capture time and to capture the genetic profile over time. This will allow us to give a lot of information about this cellular differentiation, which I told you before, and how it is affected by the different cytokines. So I'm collecting a lot of information about the location and genetic profile of different cells, of like good and bad cells responding to different cytokines. I'm using this information to selectively deliver certain cytokines to very specific cells by using like antibodies as selective transporters to only target the cells which we identify as the most beneficial. So as a summary, cells talk all the time together, and cancer cells suppress the immune system to avoid the elimination of the cancer cells. Cytokines can be used as instructive words to again reactivate these immune cells to do their job. But to design better cancer cytokine therapies, we first need more information about individual cells and also about when and where we have to instruct them. With this, I'm at the end of my talk, and I would like to thank everybody that is involved in this project from my lab. You can see here a picture from a conference, including my supervisor, Ido Amit, also Michel, Daniel, and Roberto, who are part of this project. And I, of course, would also like to thank the Azrieli Foundation for the support of this project, and also for organizing this wonderful event today, and you for your attention. Thank you.